Okay, today we're going to be talking about genetics, which is one of my favorite topics in biology. And we'll talk about the father of genetics, old Greg Mendel, and a little bit about genes and alleles and genotype and phenotype. And there's a ton of new vocabulary for this unit. So I would encourage you to make flashcards with those vocabulary terms on them. Um, at any rate, here we go. Let's start off with old Greg Mendel. Gregor Mendel was a priest who lived in a monastery, and he was responsible for the gardening there. And in his garden, he did some experiments and some observational science uh, in regards to human genetics. Now, it would have been maybe perhaps more accurate in some way if he were to use real humans, but humans take a long time to develop and a long time to reach sexual maturity. And so old Greg, he used pea plants. One, because they were readily available in the garden at the monastery, and two, because they reproduce pretty quickly. So he was able to see multiple generations of peas in a very short amount of time. So that is why he used the peas. And it's still representative of human genetics. We still follow the same trends, dominant alleles and recessive alleles, but we'll get to that in a second. Let's go back just one slide. Genetics is the study of inheritance. Okay, so I got my dad's eyes, or my mom's hair, or my mom's height. Um, so you inherit traits from your parents, one or both of them, and those traits are found in your DNA. Okay, and a section of your DNA is called a gene. A segment of DNA is called a gene. And those genes determine our characteristics, not just physical, visible characteristics, but characteristics that are detectable as well, like blood type. Okay. Now, Mendel worked with pea plants, so we're going to define some of the terms that he used, but more broadly, we will define some terms in human biology and biology of other reproducing organisms as well. So, the two human sex cells are called the sperm and the egg. Males have sperm, females have eggs, and those reproductive cells are called gametes. When two gametes join, one from a male and one from a female, when two gametes fuse, that's called fertilization. Okay. And it would be appropriate to point out that traits that are inheritable are just specific characteristics. In regards to Mendel's pea plants, he was looking at traits like whether the flower was axial or on the top, or what color the flower was, what color the seed was, whether the seed was wrinkled or round, height of the plant, several different traits, which again are specific characteristics to either a parent or an offspring. And again, traits are determined by our genes, and genes are segments of DNA. Okay. Um, there are two ways that pea plants can be pollinated. First of all, pea plants are true breeding, or can be, which means that they are asexual. So pea plants have both the female and the male parts, or pea plants can be cross pollinated, which is one plant pollinates, when one plant pollinates another plant. And their genetic material combines. So, in a sense, the pollen of a pea plant would be perhaps its gamete or its reproductive cells, as opposed to sperm and an egg in humans. When you have two different parents with contrasting characteristics, the offspring of those parents are called hybrids. Okay, so we're all hybrids. We both have, or we all have two very different parents, and we are a hybrid of those parents. And the same with the pea plants. When they have two different parents with contrasting characteristics, they're considered to be hybrids. If you look at the image there, you can see a true breeding pea plant and how cross-pollination works. Right there, you see it with a paintbrush. That's intentional cross-pollination, but sometimes bees will cause cross-pollination, other insects, the wind. So there's a bunch of different ways that plants can actually engage in cross-pollination. Some require cross-pollination, some are true breeding or self-pollinating, and some engage in both activities. If you look at this chart, these are some of the traits that Mendel was kind of tracking in his experiments. You can see he was looking at seed shape, seed color, flower color, etc. And this chart has the parent generation, which is notated with a P, and the first generation, the filial generation one. Filial stands for son or daughter in Latin. We usually just call it first generation, second generation, but the generations are notated with F and then a subscript of a number. So the first generation of a parental cross would be F subscript 1, the next generation F subscript 2, and so on and so forth. If you look at this chart, Mendel was doing some discovering, and this chart kind of shows that. Look at the first cross. A round seed parent is crossed with a wrinkled seed parent, and they ended up with just round seeded offspring. And Mendel thought, well, what's happening here? Is the wrinkled seed trait just disappearing? Is it hidden? Is it what's happening? Why do we only see one of the parent's traits in the first generation? And you see that all across the board. In the first daughter generation, we only see the trait from one parent. If you cross a green pea pod with a yellow pea pod, all the offspring were green. If you cross a purple flower with a white flower, all the offspring were purple. And Mendel was saying, well, what is happening to that other trait? Well, it takes a little bit more knowledge to understand what's going on. And Mendel actually found out what was going on when he crossed some of the first generation offspring and got the second generation, and some of those traits reappeared. How did that happen? Let's check this out. Genes are segments of DNA, we know that. And we get genes from our mother and from our father, from both of our parents. We actually have 46 chromosomes, 23 from mom, 23 from dad. And those chromosomes exist in positions 1 through 23. In the first position, there are 2 chromosomes, one from mom, one from dad. Those chromosomes contain the same genes, but there are different forms of those genes on either chromosome. So on chromosome 1, they both will code for hair color, perhaps. But perhaps the gene you got from mom codes for black hair color, and the gene you got from dad codes for brown hair color, okay? So they are different versions of the same gene. And the word to describe different versions of the same gene is allele. So alleles are just different forms of the same gene. And alleles, the different versions of the same genes, are represented by capital and lowercase letters, okay? The principle of dominance states 2 things. First of all, it states that some alleles are dominant and some alleles are recessive. And secondly, it states that when a dominant allele is present, that allele will always be expressed, okay? So if you look at the chart here, you can see 2 purple flowers and a white flower, okay? The first purple flower has a capital B and a capital B. Those are both dominant traits, or both dominant alleles. So we know that the dominant trait here, when we're talking about flower color, the dominant trait must be purple, okay? Then we look at the lowercase b right here, and you can see that it is a white flower, and it contains 2 recessive alleles, okay? Because it contains 2 recessive alleles, it will express the recessive trait, which in this case is white flower color. Then we look at the flower in the middle that is purple. It's purple even though it has one dominant allele and one recessive allele. It still only expresses that one trait. As long and according to the principle of dominance, as long as there is one dominant allele, that is the trait that will be expressed. So even though it has a recessive allele, that trait is not going to be expressed. So we still have a purple flower in this case. Where it gets interesting is that purple flower has the allele that would cause for the flower to be white. So if this particular purple flower bred with another flower that had the recessive allele, there's potential that it might perhaps produce a white flower, even though it was purple and its mate was purple. Or if it mated with a white flower, it might also have the probability of producing a white flower, and at a greater percentage, because there would be more opportunity for those two recessive alleles to join together. Okay. Now we get to kind of classify these combinations of alleles. So if you look at the left on that image of the flowers, you'll see capital B, capital B. That's two dominant alleles. When we have two dominant alleles, it is said to be homozygous dominant. Now, the root word homo means same, so we have two of the same alleles, and dominant tells us that we have two of the same dominant alleles. So capital B, purple, purple flower. Then we look to the right and we see homozygous recessive. That means we have two of the same, homo, and recessive, so two recessive alleles. So that combination of alleles is called homozygous recessive. Then when you look in the middle, we see heterozygous. The root word hetero means different. We have two different alleles, one dominant and one recessive. So we call that combination of alleles heterozygous. Now, when we're looking at the physical traits that are expressed and the genetic makeup, whether or not it's homozygous or heterozygous, there are words for that. So if I want to know what the genetic makeup is of this purple flower in the middle, I would ask, what's the genotype? Okay, so the genotype for this purple flower in the middle is actually heterozygous, or heterozygous. Yeah, the genotype is heterozygous, big B, little b. When I ask for the genotype, or when you're asked for the genotype, typically you will give the expression in the form of capital or lowercase letters. But you could accurately answer what's the genotype. You could say it's a heterozygous genotype, which we would know then it would be a capital and a lowercase letter, one dominant allele, one recessive allele. The phenotype is what we're actually seeing, the physical trait. Now, I say that with a grain of salt because sometimes the phenotype is not something visible right off the bat. Like the phenotype could be blood type. I can't see the blood type, but I can certainly do a test and figure out the blood type. So I like to say that the phenotype is the detectable trait, okay, the trait that we can detect. Every organism has a genetic makeup and a physical trait, or a detectable trait. So every organism has a genotype and a phenotype. The best way I can describe this is the genotype is the genes and the phenotype is what we physically see. Again, physically with a grain of salt, because sometimes we can't actually see the trait in reference, like blood type. Segregation is when the alleles are separated during gamete formation, okay? During gamete formation, the alleles for each gene segregates from each other so that each gamete carries only one allele for each gene. This can be better exemplified in the next image, where we see that we have two parents in the F1 generation, okay? So their genotypes are big G, little g, big G, little g, heterozygous, dominant and recessive, alleles in both parents. So in our first parent on the left, we see that those alleles are separated or segregated when the gametes are formed. Now we'll go on to a whole other unit about meiosis, where we see the very complex way that these gametes are separated. But for now, we just need to know that our gametes are our parent genotype. The alleles will be separated during gamete formation. So we see that the first parent is heterozygous. It's got a dominant allele and a recessive allele. Well, when those gametes are formed, the chromosomes are cut in half or divided in half, and only one allele ends up in each gamete, in each sex cell, in each sperm or egg. So we see that we've separated our two parent genotypes into four separate uh, gametes here. And those gametes can recombine in a multitude of ways. We can see here that we started with two heterozygous parents, and we end up with a homozygous dominant child, two heterozygous children, and one homozygous recessive child. And this is how this particular example, when we breed the first generation to get the second generation, this is where Mendel was like, oh, the recessive alleles don't disappear, they stay there. And in the event that two recessive alleles are combined again in a subsequent generation, we then see that trait again. So this kind of solidified Mendel's understanding of dominant and recessive alleles and where they go. And he just knew now that the recessive alleles didn't disappear. They were still carried on, but we needed the presence of two recessive alleles for that particular trait to occur. When we're looking at a chart like this, or when we do a Punnett square, or when we are predicting what the offspring of two parents will look like, we're talking about probability, okay? If I flip a coin, the probability that I'll get heads is 50%. But if I flip a coin 3 times, the probability that I'll get heads every single time is only 12.5%, right? So it's the same thing with genetics. We could have gotten heads every single time, but the probability was very low. We could have gotten tails every single time, but the probability was very low. The probability really is kind of 50-50. Is it heads or tails every single time? And it's the same with genetics. Here we see the example of the two green parents producing either a green offspring or a yellow offspring. We can see that it's only a 25% chance that they will produce a yellow offspring. But theoretically, they could produce all yellow offspring, okay? We're just talking about probability. What's the likelihood that they would produce that type of offspring, okay? In this situation, there's a 75% chance that they will produce a green offspring, and there's a 25% chance that they will produce a yellow offspring. Mendel noticed that when he crossed two plants that were hybrids for pod color, about 3/4 of the resulting plants had green pods, and about a fourth had 3/4 had green, and about a fourth had yellow. We can determine the probability of certain traits being passed on to offspring through the use of a diagram called the Punnett square. Okay, and the Punnett square gives us the probability that a certain genotype or phenotype will occur. You can see in this example that we've crossed a green homozygous recessive pea pod with a heterozygous pea pod. And the result is 50% of the offspring were yellow, heterozygous yellow, and 50% of the offspring were green, homozygous recessive green. So we can now see the likelihood of these two parents. It's 50% probable that they will have yellow offspring, and there's a 50% chance that they will have green offspring. They could have all yellow, they could have all green. We're just talking about the probability that this would happen. If they had 4 kids, it's likely that half of them would be yellow and half of them would be green. Now, we'll have more practice with Punnett squares in class, but I do want to let you know how they work. What we do is we draw a box and we separate it into 4 chambers, 4 squares within one single box. We take both of our parent genotypes and we put one allele on top of each row, and we put one allele on the side of each column, typically to the left. And then each box will get an allele from the top and allele from the side. So you can see in this first box here, we get the dominant allele from the top and the recessive allele from the side. And that produces this heterozygous genotype, big T, little t. And we do the same for all the other boxes. One from the top, one from the side, one from the top, one from the side, one from the top, one from the side, okay? It's kind of difficult to see here, but when we do examples in class, it'll make a lot more sense. And we see here, if you cross a homozygous dominant genotype with a homozygous recessive genotype, we end up with 100% heterozygous. So all of the offspring will express the dominant trait. Now that I've told you all the rules, I'm going to tell you how the rules get broken. Okay, the first example is incomplete dominance. Okay, sometimes the alleles are not necessarily completely dominant or completely recessive. When this happens, it's called incomplete dominance. In a scenario where we have incomplete dominance, the phenotype is actually a blend of the new phenotype is actually a blend of the two original phenotypes, okay? So in this example, the homozygous dominant phenotype is red flowers, and the homozygous recessive phenotype is white flowers. Now, when we get a heterozygote, an organism or a flower in this case that has both the dominant and the recessive allele, we end up with a brand new phenotype, which is a combination of the two alleles, okay? So the combination of red and white is pink in this case. And sometimes they don't blend, but actually both traits are expressed. When both traits are expressed, this is called codominance. Codominance. When we have two alleles that end up being clearly expressed, it's called codominance. So, for example, in some chickens, if we would breed a black chicken with a white chicken, we might end up with a black and white chicken. Both alleles, both traits, both the dominant and recessive traits, white and black color, would be expressed. And you can see that in this Dalmatian chicken over here, both white and black. So when we get a heterozygous genotype, both alleles will be expressed. Instead of blended together to create a new phenotype, both of the original phenotypes are expressed. In a situation where we're talking about these chickens, if it were incomplete dominance, we wouldn't have both white and black feathers expressed. We would get a blend of the two to create a new phenotype. And if we were to blend black and white, we would end up with gray. So if this trait were incomplete dominance instead of codominance, we'd get a gray chicken. But in this situation, it is codominant. And then we come to sex-linked traits. And I'm glad that we have this image over here. You've heard me say 23 chromosomes, 46 chromosomes, 23, 46. Well, this is a good way to look at how our chromosomes look when all of our cells have 46 chromosomes. And there are 23 positions for those chromosomes. And in each position, there are two chromosomes, one from mom, one from dad. And those chromosomes in each position hold the same genes, but they are alleles, right? They're the same type of gene, but they're a different version. So for example, chromosome number 2 might code for hair color, but there's two chromosomes in that position. So one might code for black hair, one might code for blonde hair. So remember, alleles are different versions of the same gene, and this is a great example of how to kind of visualize that. The 23rd chromosomes are considered to be sex chromosomes, and they're called that because they actually determine the sex of an organism. If you have two X chromosomes, you're a female. If you have an X and a Y chromosome, you are a male, biologically. There are certain traits that end up on these sex chromosomes, most frequently the X chromosome because it's bigger and has more genes. But when there are traits that are on the sex chromosomes, they're called sex-linked traits. And sex-linked traits are actually notated a little differently, even though they behave similarly to other traits. It just so happens that they're on the sex cells or the sex chromosomes. But they're notated with a superscript above an X, okay? So if you'll look right here, you can see that there's an X with a capital H superscript. So that would be the dominant allele. Instead of just writing a capital H, we do an X with a capital H superscript. Or we could do the recessive allele and have a lowercase h there, okay? So we've covered a lot in this lecture. Genetics is much more complex. We've just scratched the surface, but this gives you an idea of the new vocabulary, what we're supposed to be learning in class, and hopefully will serve as a source for you to review if you missed the lecture in class. Okay, very good. I will see you at the next lecture.